My name is Evencia Sanchez Mejias, and I'm CEO and co-founder of Integra Therapeutics. 15 years ago, when I started working in clinical genetics, gene therapies to cure patients with genetic conditions was a distant dream. Today, these therapies are a reality, but there is still a lot of work to do. And I will tell you how we are writing the future of gene therapy. Integra was born in 2021 in Barcelona with the goal of accelerating the development and broaden the scope of gene therapies. My CSO was founding scientist of eGenesis, a gene writing company based in Boston that has raised over 200 million, and co-founder of S-Biomedic, which exited to pharma last year. Together, we secured the largest ever biotech company creation round in Spain. Today, we are advised by world-class scientists, including Professor George Church, who is a pioneer in synthetic biology, and we have grown our team to almost 20 people. We have made significant progress in solving key limitations in gene therapy. Specifically, there are three aspects of the current technologies for gene editing that doesn't allow this to be applied to a wider spectrum of patients. Current gene editing technology lacks size scalability for larger payloads. With the current tools, there is no control over the insertional site, which can be harmful to patients. And finally, gold standard for gene therapy, the AAV vectors, lack durability of expression, limiting its application and long-term benefits. To overcome these limitations, we created FICAT, a fine can't transfer gene writing platform, to unlock new horizons in gene therapy. FICAT combines the programmability of RNA-guided nucleases with the efficiency of large DNA integration capacity of transposases to perform precise integration of therapeutic genes of virtually any size. The RNA-guided nuclease CRISPR does the targeting to a specific site in the genome and performs a double standard break that is then used by the transposase to integrate the therapeutic payload in this site. Through protein engineering and synthetic evolution, we have developed FICAT, and we are proud to say that this is the first genome engineering tool that provides programmable DNA transposition with no off-target integration of therapeutic genes of virtually any size. We have been able to validate these in ex vivo settings for up to 22 kbs and in vivo for up to 11 kbs, with precision and no off-target, and FICAT works in dividing and non-dividing cells, so the expression of the gene is not diluted with cell division. During the last 2 years, we have been focused on FICAT platform development and adaptation for advanced therapies. We will continue to be at the forefront of gene writing technology, and we are now ready to develop our first therapeutic product. Integra will focus in developing an LNP-based in vivo gene therapy with our lead indication reaching IND at the end of 2026. We will also partner with pharmaceutical and biotech companies in this space for the development of cell-based therapies. Rare diseases are not rare at all collectively. There are 350 million people affected by these conditions, and these patients represent a larger economic burden to the healthcare system than the mass market patients. There is only 5% of diseases in which there is treatment, mostly addressing the symptoms and not the underlying cause. And FICAT can significantly impact these patients, families, and communities, providing cures to last a lifetime. We first focus on liver monogenic diseases to the risk for the delivery aspects, as LNPs are excellent for liver delivery. So we can focus on demonstrating FICAT efficacy and safety. We have a screen for 100 indication in this space to find the one that best placed to FICAT strength, and selected an undisclosed pediatric liver indication with congenital onset, high medical need, clear biology, that is a perfect fit for FICAT competitive advantages. To cure our lead indication, there is required the delivery of a large transgene, the sustained expression throughout the patient's life, and precise control on the insertion site. Our technology is best suited to address these challenges, and Integra could potentially be the first to provide a curative treatment for this devastating disease. We are currently fundraising to continue the development of our therapeutic products, and you can join us in bringing FICAD closer to patients. Thank you.
Aventia, thank you so much for the pitch, just on time. And we're waiting for our jury to come on the virtual stage. And the question to you is, what are actually the challenges and opportunities of running a startup?
So it has been a huge learning progress. I'm a scientist by training and now managing this company. So I'm very thankful for all the formal and informal mentors here in the Catalonian biotech community and outside for all the support. And yes, there are many challenges transitioning from doing academic science to managing a biotech company. But so far, it has been a great journey.
Thank you, Aventia. So maybe can you speak a little bit about the in vivo work, also then a little bit about the delivery topic that you mentioned, go to what we've been seeing more recently, but I assume that given that you've outlined, you would assume that there's kind of opportunity delivery strategies.
Apologies, we don't hear you well. Something is with your audio. Can you mute yourself and then unmute yourself once again?
But I think I heard the question. I think I heard the question partially. So the question is about the in vivo program and the delivery and what technology we are using for this. Perfect. So yes, we strategically we decided to stay away from viral vectors, not only to take advantage of the large payload capability that FICAD has, but also for safety reasons in order to do systemic delivery of our solutions. So staying out of viral vectors, we decided to go for lipid nanoparticles, which are well tolerated and can target the liver relatively easy. We can think of expanding to other tissues as the viral-free technology becomes mature. But for the moment, we are using LNPs to target the liver. And this was a big challenge because we had to do both mRNA and DNA. And we have successfully developed the DNA delivery in a viral-free manner for our solutions.
Thank you.
No problem. And Fabrizio?
Thank you. So a comment on the question. The question is about the competitive advantage in this very crowded space. And then I really appreciated the idea of mixing a platform and a proprietary pipeline. That makes a lot of sense. Could you elaborate a bit more on this? Thank you.
Yeah. So for competitive advantage in the gene writing space, probably for 5 minutes we wouldn't have enough. But you're right that these gene writing tools are maturing very fast. And I think we have a unique opportunity with our IP position and also the simplicity of the system compared to what others are bringing to the space. But for sure, it's a challenge and an opportunity to be in this gene writing space, and we have to think very smart on the platform and the pipeline at the same time to be able to compete with others in the space. And then regarding the dual business model of proprietary products that we can fully develop in-house, retain the value of the platform in the company, and also because we cannot do it all and this tool can serve for other things, we decided to partner for the ex vivo space, in which we don't develop our own cars, for example, and there are many other players outside which could need greater cargo capabilities or more precision, and we can partner with others that are closer to the clinical stage and get a second stream of revenue that we can deploy in our internal pipeline. And Tugrul?
Right. Well, thank you very much for the nice presentation. Obviously, targeting liver is kind of a safer route for you, but I can't stop thinking which monogenic pediatric liver disease are you planning to select that would require a large cargo to be delivered that is not crowded, where the standard of care is inefficient?
Yeah.
What's your strategy for selecting a program?
Yeah. So at the moment, we are not ready to publicly disclose our pipeline. But yes, this was probably one of the most strategic decisions that we have to take at Integra, which is the first, and then the expansion of the pipeline. We wanted to have as less risk as possible in the development. That's why we selected the liver and also a rare indication, orphan disease, etc. But we wanted this first product to have also commercial value and be of interest. So we stay away of other things that are closer to the market or even already approved, such as hemophilia, for example, and selected an indication that is not crowded in terms of competitive landscape. We could potentially be the first ones reaching a market. And there is a number of patients that will allow not only for the clinical trial, but also for the commercialization thereafter. Is in the rare disease space. So this is something that it's not going to be a blockbuster for the first indication, but will allow us to also expand the pipeline. So that was the rationale. And I know I haven't replied your question, but this is as much as we can say.